Good evening, everyone. Hello, and welcome to the first Arboricultural Association webinar of 2026. My name's John Parker, CEO at the Association and your host for this evening. Please do say hello in the chat and let us know where you're watching from. This evening, we've got two great talks all the way from the US of A. We'll be hearing about the golden oyster mushroom with Michelle Jusino and responses of ectomycorrhizal fungi to elevated soil salinity with Elena Karlsen-Ayala. That will be in just a moment, but first of all, some news about the remaining webinars of the series. Next week, on January the 14th, Jim Chambers will be navigating the many definitions of ancient trees. There'll be sure to be a bit of controversy in that one. On January the 21st, Harry Studholme will tell us about reforesting Devon. On January the 28th, it will be all about bats and trees with Jim Mulholland. And then on February the 4th, Tony Kirkham and Kenton Rogers will be presenting their new book, the latest publication from the Arbor Association, The Tree Care Manual. And then on February the 11th, it will be the final webinar of the series with Ted Green in the company of Arbs. And you can sign up for all of those and catch up with any webinars you may have missed on our website. As ever, as I've already said, but it's worth remembering, please do say hello in the chat. But if you have a question, please use the question button. Don't put it in the chat, otherwise I'll lose it. I won't see your question. It'll be a wasted question. You'll be asking questions into the void. It won't be answered. So please use the question button. And please do say which of our speakers you are aiming your question at. Otherwise, it'll also cause terrible confusion. I'm just a simple man. So it is now my very great pleasure to welcome our first speaker to the virtual stage. Elena, over to you.
Good evening, everyone. Good afternoon, good morning, depending on where you are in the world. Thank you so much to the organizers for the invitation to talk to you today. My name is Elena Karlsen-Ayala. I am a research plant pathologist with the Forest Service in the United States, and I'm located in Connecticut, which is a state close to New York City. So today I'm going to be talking about responses of ectomycorrhizal fungi to elevated soil salinity. So a lot of the research that I'm interested in is really focused on sort of this central question of how do disturbances impact microbial communities, and how do disturbances lead to the loss of microbial functional diversity and tree host resilience. And so for the sake of today's talk, the disturbance that I'll be focused on is increased salinity. But these disturbances can be broad, and certainly in the face of changing climate conditions, sometimes these disturbances are coupled with other disturbances. So just for like a ecology 101, I want to discuss the difference between pulse versus ramp disturbances. So pulse disturbances are usually sort of a short-term disturbance that can happen kind of in one go. So that could be things like hurricanes or the impacts of salt spray from hurricanes, wildfires, floods, etc. Whereas ramp disturbances are sort of disturbances that occur over time and sort of accumulate as a result. So these could be things like drought, sea level rise, salt intrusion as a result of sea level rise, irrigation practices that increased soil salinity, or climate warming. And so one example of sort of the impact of a ramp disturbance of having increased soil salinization or increased salinity into ecosystems is ghost forests. So ghost forests are these phenomena of sort of standing dead trees in areas that have become uninhabitable for those trees to survive. And usually that's a result of saltwater intrusion or increased salinity in that ecosystem. And so ghost forests are very common in the United States, in particular in states that have coastal forests. So these could be states like Florida, Georgia, and North Carolina. And it's definitely a growing concern because of the increased salinization as a result of sea level rise and salt intrusion. And so for today's talk, I'm going to focus on Florida as sort of a case study of this increased salinity in ecosystems and a place where there are known ghost forests. So Florida is a pretty low-lying state. It's located in the southern range of the United States, and especially southern Florida is incredibly low-lying, with the vast majority of this ecosystem less than 2 meters above sea level. And so Florida is also sort of a great example for increased salinization, because there's a lot of disturbances that are pretty inescapable in this region. One of them being hurricanes and the threat of sea level rise in these low-lying environments. And so for my PhD research, I focused on this really unique ecosystem called the South Florida pine rocklands. This is an ecosystem that is endemic to southern Florida. So you can see this is Miami-Dade County, which is sort of the tip of Florida, and this ecosystem is also found in the Florida Keys, which are islands off of the coast of Florida. And so in this figure on the left, you'll see that in green, we have the historic range of this ecosystem outlined, but in teal, we have the current range. So this portion right here, which is sort of the largest continuous section of the ecosystem, is within the Everglades National Park, which is a protected area. But outside the Everglades National Park, we can see that less than 2% of this ecosystem still remains. So it's a highly fragmented ecosystem. And that's particularly because of, you know, the influence of urbanization and habitat destruction for development. And so because this ecosystem is only found in southern Florida, it hosts a multitude of insects and animals that are endemic to this ecosystem. So it's a really important ecosystem for this state. And it's pretty unique in that it's characterized by this open canopy of South Florida slash pine, and it's a fire maintained ecosystem. So the pines in this ecosystem are a keystone tree, and their role is to provide fine fuels to help the system maintain sort of low intensity fires every 3 to 7 years to prevent the overgrowth of tropical hammock plants that create a closed canopy ecosystem. And just to sort of continue to show how unique this ecosystem is, it's sort of aptly named the pine rocklands because the ecosystem literally grows out of this oolitic limestone, which is sort of a byproduct of the geological history of this area that, you know, historically was underwater. And so this karst geology makes for, you know, a pretty difficult ecosystem to persist in. And so, as I mentioned before, there's a lot of habitat fragmentation and destruction in the area. And so there's been a lot of local efforts to restore this ecosystem by planting, planting native plants and trying to restore it. However, prior to my PhD research, there had been really no studies looking at the role of ectomycorrhizal fungi in these environments. And I'll get into the importance of that in a minute. But I just want to show you again the importance of these keystone trees and to explain that historically there's been a lot of difficulty in pine seedling recruitment and in mature trees, dying during hurricane activities and then having trouble getting new seedlings to establish so the ecosystem can persist. So ectomycorrhizal fungi. So the vast majority of plants associate with some sort of mycorrhizal fungi. Mycorrhiza meaning fungus root. So myco is fungus, rhiza is root. The two predominant types of mycorrhizal fungi are ectomycorrhizal fungi and arbuscular mycorrhizal fungi. We're going to be focusing on ECM today because that's the association with pines. But these mycorrhizal associations are a mutualistic relationship between plants and fungi, where the plants are providing carbon as a byproduct of photosynthesis to the fungi, and the fungi are sort of scavenging nutrients in the soil and secreting enzymes that help mobilize the nutrients for the plants to uptake. And so because the mycorrhizae can convert these fungi into, can convert sorry, let me take a step back. The fungi mobilize nutrients in the soil, and these nutrients are then usable for the plant. So this helps improve nutrient uptake. And one of the sort of important components about mycorrhizae is that they can help improve stress tolerant in the plant. So this can be tolerance to things like drought and heavy metals, and to provide some protection against diseases. And so a lot of, not all, but many of the fungi that you find fruiting in the forest can be ectomycorrhizal fungi. Some of them are very prized edibles, like Cantharellus and tuber. And basically, ECM fungi provide this fungal sheath around the outside of the plant root, which forms these really charismatic, cute fungal morphotypes that you see here in this photo in the middle. And just to sort of make this point really clear, a lot of trees, particularly pine trees, are obligately associated with these fungi. So the picture down here shows pine seedlings grown with ECM fungi and without fungi. And so you can see they're very, very important. So just to kind of make this point clear, ECM fungi are critical to the establishment and survival of pines. They had not previously been studied in restoration efforts in the pine rocklands, and there's been this long history of poor pine seedling recruitment and loss of mature pine trees. And so I started to think about the fact that this ecosystem is pretty salty. And so through some of the other work that I did in my PhD, I sort of noticed that along the range of this ecosystem, this point up here being the northernmost range of the ecosystem, all the way down to the Keys, which is the southernmost point in this ecosystem, there was sort of a natural soil salinity gradient. So when we look at soil EC, or electrical conductivity, the total salts in the soil, or we look at magnesium, sodium, or calcium, we can see that as we go further south, the soil salinity increases. And particularly when we look at sodium, we can see that in the southern range of this ecosystem, the soils were 34 times saltier than in the northern range. And so this certainly got me thinking, this is probably influencing pine seedling recruitment and the mature trees as well. And there's certainly a history of research in ECM fungi and salt. Generally speaking, though, most studies that look at ECM fungi and salt focus more on the plant response to having ECM fungi associated with it. So what that means is studies may take specific ectomycorrhizal fungi, inoculate the roots of those trees, and then sort of assess how the trees fare under high salt stress conditions. And we certainly have examples of ECM fungi that grow with trees in high saline environments. So this photo up here is a cocolova tree or a sea grape tree. These are trees that are often found on beaches, so very salty areas. And fungi like scleroderma are known to help protect against salt stress. And there are certainly taxa that have been studied. This is not an exhaustive list of fungi, but there are many fungi that have been studied for their role in salt tolerance. And this is fungi like Hebeloma, Laccaria, Rhizopogon, Pseuilus, etc. However, the majority of ECM fungi are not considered salt tolerant. So most of the research looking at salt and ECM either looks at particular seedlings that are inoculated with particular focal species of fungi, or they'll look at mature pine trees in high salt areas and look at the fungal community composition associated with those trees. But for the sake of this talk, we're going to be focusing on this paper that I published titled Salt Life: Salinity Drives Ectomycorrhizal Community Structure in the Endangered Pine Rocklands. And for this study, which we published last year in Molecular Ecology, this is one of the few papers that has combined both surveys of the ectomycorrhizal communities associated with mature trees, followed by seedling inoculations and salinity manipulations, and then looking at the total community response of the total ECM community response to these salts. And so the two driving questions of this research is, how is soil salinity already driving differences in ECM fungi communities among mature trees? And how do changes in salinity explain any of the community structure and diversity of seedling ECM community composition? So for the first part of this research, we wanted to survey mature pine trees. And so far, before this research had been done, there was no work looking at the community structure of mature trees at all in this ecosystem. So it was pretty cool to sort of have a first look at what communities were associated with these trees. So we selected 8 sites, again, ranging from the northern part of this range in orange, the central part of this range in purple, and then the southern part of this range in blue. So we selected 8 sites, and we sampled 10 trees per site by collecting roots at the base of a mature pine tree. Now, what I'm going to show you here is a constrained correspondence analysis. This is basically a way of taking multivariate data and condensing it into 2D space. And so each point on this figure corresponds to the roots of a mature pine tree. And as you can see, the closer these points are, the more similar those ECM communities are to each other, and the more spread apart the points are, the more dissimilar they are. And so what I want to focus on here is we can see really nice clustering. These 3 ordi-ellipses show really nice clustering of the 3 ranges. So the orange range being the northern part of the forest, the blue being the southern range, and the purple being that central range. And so we see really distinct clustering in the community composition. We can see that elevation is statistically significant in driving the community composition in those more northern sites. And we can see that EC, or electrical conductivity, the total salts in the soil, are significantly and positively correlated with the more southern sites, and elevation is negatively correlated with the southern sites. And then we can see for these central sites, distance from the ocean and pH drive those communities. Now, when we look, so this figure was looking at the total ECM community composition. Now, when we look at OTU richness, this is operational taxonomic units, which you can think of, OTU richness, you can think of how many different OTUs we found associated with these trees. When we do a correlation between electrical conductivity and OTU richness, we can see that OTU richness is significantly negatively correlated with electrical conductivity. So the higher salts in the soil, the fewer OTUs we were finding. We also found that elevation was significantly positively associated with OTU richness. So these lower elevation sites in the south had fewer OTUs than in the north. And what is really particularly important about elevation in the context of this study is that we're talking about very, very small elevational ranges. The vast majority of we're really only talking about a 5-meter difference in elevation here. And so certainly there are other studies that have looked at ECM community composition across elevational gradients, but that's usually, across hundreds or thousands of meters. So this is pretty impressive that really small changes in elevation have significant impacts on OTU richness. Now, when we look at distance to the ocean and we correlate that with OTU richness. We did not find any statistical significant difference. Same with soil pH, and also same with size of habitat patch. So all three of these variables were not significantly correlated with OTU richness. And the soil pH and size of the habitat patch were sort of surprising to us, because these are two variables that we would have expected to be significantly correlated with OTU richness. Now, switching gears to the second half of this experiment, this is the manipulative salt experiment. So again, along this range of the ecosystem, we selected 3 of the sites that we had already selected for the mature pine trees, but we then selected additional mature trees, and at the base of each tree we put a quadrant down. We collected soil at the base of those trees that would be used in our greenhouse study. So just to reiterate, we had LPT, Larry and Penny Thompson, is our northern range in Miami. Navy Wells Preserve, or NWP, is in Homestead, and then the National Key Deer Refuge, Big Pine Key, is our southern range. And so we had 10 trees from these 3 sites, gave us 30 soils. So we had the 30 soils. We then had 4 salt treatments at 0, 25, 50, 75 millimolar. And we had 5 seedling replicates of each of those, for a total of 600 seedlings, which we grew in the greenhouse for about 8 months. Then at the end of the experiment, we picked off, these are the seedling root tips, and you can see here the ectomycorrhizal root tips. So we washed the roots, we picked off all of those ECM morphotypes, and then for the 5 seedling replicates, we combined all of those replicates into one pool for each of the 30 soils by the 4 salt treatments. And so that gave us 120 samples, or 120 pools of fungi. We then sequenced these fungi using Illumina-MySeq of the fungal ITS1. And we did that for the mature trees. Sorry, the order of the slides, I didn't mention that, but yeah, the mature trees, we also did Illumina-MySeq of the fungal ITS1. So prior to the experiment, we sequenced the total fungi in the soil before we planted the seedlings and did the salt treatment. And again, I just want to show you, this is another constrained correspondence analysis, and we see the same pattern where we see our northern sites and our central sites and our southern sites clustering differently. Now, what you'll notice here is you can see that there's sort of differences in the number of points for each of these regions. And this is really interesting in that some of our sites, particularly our southernmost sites, were really, really salty. And because of how much salt there was in the baseline soil, we had difficulty with PCR amplification. So it kind of goes to show that, you know, EC, total salts in the soil, were really problematic. And so maybe unsurprising, we can see that electrical conductivity is really driving the community composition in these soils. And then distance to the ocean was really driving the central sites. So at the end of our experiment, we had some seedlings that did not survive the experiment. So we had no mortality in our control seedlings that were watered with just water. In our low and medium treatments, we had 6 seedlings that died combined, and these were only found from the southern sites. And then for the high seedling, high salt treatment, we had 16 from the southern range, 6 from the central range, and 5 from the northern range that died. And so what we can see here is that, especially for the southernmost range, those very salty sites, even really small increases in salt had detrimental impacts on these seedling communities. So just an important note here, and may help to explain why we have a lot of seedling mortality during restoration or just natural seedling recruitment. At the end of the experiment, we measured the electrical conductivity, the total salts in the soils, as well as the soil pH. And what we did find is significant differences in electrical conductivity with, the controls being very low, and then low, medium, and high salt treatments having this gradual increase in soil salinity. This is not surprising. We sort of would expect this since we've been watering the plants. But what this does show is that the salts do accumulate in the soils over time. But what was sort of interesting is despite that high salt treatments, we really didn't see changes in the soil pH at all, no significant differences there. And when we look at the total seedling fungal communities, again, this is a constrained correspondence analysis. Here we're using electrical conductivity, soil pH, and treatment effect as variables in our model selection. And we find that electrical conductivity and treatment are significant drivers in the community composition, and that electrical conductivity is a significant driver, particularly in the high seedling communities. And so again, I just want to point out that we saw a very strong pattern here, despite having some seedlings that died throughout the experiment and therefore do not have fungi that would be put into this analysis. And so even having some seedlings die throughout the experiment, we still found this really strong signal throughout. Okay, so before I explain one of the last figures in the paper, I want to just take a step back and mention ECM lineages, just to have some framework to understand that last figure. So when I talk about ECM lineages, we're talking about distinct phylogenetic evolutionary groups of fungi. So ECM, this ectomycorrhizal guild in the kingdom of fungi, has evolved multiple times throughout history. And so when we want to think about how different lineages of fungi or different groups that have the same phylogenetic origin, we can think about different traits that those groups have. So one lineage is the Rhizopogon sewillis group. This is pine specific, basidiomycetes, and long distance exploration types, which I'll get to on the next slide. We have things like the Cenococcum lineage. This is an ascomycete fungi, a contact exploration type. It's known for having these melanized spores and has some ability to tolerate stress, likely because of that. And then we have groups like Tomentella tholoptera, which are basidiomycetes, contact, and short distant types. And so when we think about lineages, they often have, even within a same lineage, so a same evolutionary history, there might be traits that are different among the fungi. So in this Rhizopogon sewillis clade, we see Rhizopogon are these sequestrate truffle-like fungi, whereas sewillis are sort of your more typical agaricoid, mushroom-forming body. But they share sort of similar functional traits, where that could be exploration type, the metabolic activity. And so grouping these into lineages can help us understand how these different genera of fungi sort of operate in an ecological framework. And as I mentioned, exploration types, so early on when I talked about ECM fungi, I discussed that the plant is providing carbon to the fungi. Well, different types of fungi have different carbon requirements from the tree host, depending on sort of the traits in terms of their enzymatic capabilities, in terms of how much mycelium they produce. So contact exploration type are these fungi that stay very close to the tree root. They scavenge for nutrients very close to the root. And so they don't require a lot of energy from the host. Whereas long distance exploration types tend to produce, ample amounts of mycelium, sometimes rhizomorphs that sort of help with this long distance exploration, but they can be very carbon intensive. And so this is just to explain what I meant on the previous slide about exploration types. So one of the questions, that sort of drives this research is how functional diversity is changing under these disturbances. So in this figure here, we see different OTU lineages. You'll notice that all of the guilds in purple are basidiomycetes and all in green are ascomycetes. And then all of the taxa that are outlined in this box are contact exploration types, whereas the others are more of these medium or long distance. And so what I was sort of interested in is, as salt increases, what is happening to the functional diversity? What's happening to these lineages? And so we can see in the instance of the more northern range that as salt increases, we have sort of a shift towards more ascomycete fungi, more contact exploration types. We see that less so in the central range. We do see some increase in the contact exploration types, but it's sort of staying the same from, compared to low and medium. But then in the southern range, we don't see that pattern. We see that as salt increases, we basically go to only one type of guild, which is the Rhizopogon psilolis guild. And so what we're seeing is in some of these sites, especially the high salt sites, we're seeing loss of functional diversity in those higher salt treatments. And so there's sort of two potential hypotheses here in terms of that shift. So either when the plants are exposed to higher salinity, the plants are stressed and therefore there's less carbon available, which sort of makes that shift towards the contact exploration types in the northern and central soils. Or it could just be that long distance exploration types just happen to die quicker in the study, and that's just the pattern that we see. However, that is not the case that we saw with the southern site. And what has sort of been interesting about all the work that has been done throughout my dissertation research is that over and over again, whenever we did studies with seedlings, we almost always found Rhizopogon to be pretty much the only fungus associated with these seedlings. And we can see that in these really high salt treatments, this particular functional group basically goes to fixation, with some of these seedlings only having one type of Rhizopogon on their seedling. And so certainly because of this location of these southern sites being in islands, we can certainly expect that island biogeography might be playing a role in here. So there's a lot of isolation from the mainland, and therefore there might be lower diversity here. However, one thing that, I think is probably driving this pattern is that that salt is sort of acting as a selection pressure in this ecosystem. And so perhaps the population of Rhizopogon in this ecosystem have developed more salt tolerance in comparison to fungi on the mainland. And so if that were the case, this could be a great avenue to try to isolate these fungi and use them in inoculations to then use for outplanting these seedlings in restoration. And so some of the future work would be, looking at these sort of genomic differences, genomic signatures of the Rhizopogon in the keys to see if there are genetic differences. We also would need additional studies to answer questions such as, does high salinity inhibit or kill the ECM fungi? Does high salinity mediate competition between ECM fungi, resulting in lower diversity of seedlings? And does high salinity impact carbon fixation of host plants, which then subsequently impacts the ECM communities? Or is it some combination of all of them? And so what I hope to leave you with is, the pine rocklands are really this unique ecosystem that is sort of a case study of, what could happen in a low elevation ecosystem that houses a lot of critically endangered insects and plants and animals that rely on this ecosystem. And it sort of shows us that in all of the different analyses that we did, we saw that soil salinity was really the most significant variable driving these communities, which sort of suggests that the influence of salt is already having impacts on the ecology of these fungi, and that as salt increases in this ecosystem, this could be really detrimental to the ecosystem in general. My computer froze. Can you see the next? Oh, there we go. But what I want to leave you with, since I know we've got a very global audience on the call today, is that, the pine rocklands in Florida is just one snapshot and one case study, but there are many, many, many low elevation coastal zones that harbor really ecologically important forests all around the world. And when I talk about forests in these low elevation sites, I'm not just talking about mangroves and salt marshes and ecosystems that are accustomed to high salinity, but I'm really talking about pine forests and oak forests and beech forests and other sort of forested trees that are not salt tolerant, that persist in these low elevation areas. And so we can see in the blue, these are all low elevation coastal zones. The black dots represent islands that are less than 10 meters above sea level. So lots of areas for potential research. And I wish that I had a map where I could overlay sort of low elevation zones with those really important forested areas, but couldn't find a map that showed that. But what I do want to leave you with is there are many, many unanswered questions about the influence of mycorrhizal fungi and trees in these low elevation, high disturbance ecosystems. And I hope that my talk is sort of a call to arms to study these ecosystems before they potentially disappear, and to help us understand how to restore and preserve other ecosystems elsewhere. So with that, I just want to thank my university and some funding sources, and I'll leave it at that. Thank you so much.
That was fascinating. Thank you so much, Elena. I've had to change venue. If anyone's asking why my wall color has changed, my Wi-Fi was dropping out, and I'm extremely paranoid after previous webinars we've had where that's been a problem. That was brilliant. Thank you very much. And loads more research to be done. We like it when it's left with lots more research to be done. So that means you can come back and tell us when you've fixed the problem across the globe. So we're looking forward to that. Please do keep putting your questions in for Elena. If you've got questions, please do make sure you say who they're to, because sometimes it makes sense to you when you're asking the question, but like half an hour later, it doesn't make any sense to me when I've got to read it out. So try and make that clear. Thank you, Elena. I will now hand over to our next presenter, who I believe is with us. People are raising their hands. Don't raise your hands. It's not going to work. It's not going to work. I'm not going to come to you. I'm not going to let you present. You quit and reopen. Elena, do you want to come back for a second?
I will be right back.
I've got some questions I could ask you while we're setting this up. Otherwise, it's just me and the dog. There's Elena. Brilliant.
I'm here.
It's seamless. See, it seems like stuff goes wrong, but it hasn't really gone wrong. It's all fine. I will ask you a couple of questions while Michelle is rejoining. Gavin's asked, what type of salt water was used in the irrigation? Was it seawater or was it normal water with added salts?
Yeah, so we actually used, we got aquarium. So when you have aquariums, you can buy aquarium salt, which is basically mimicking seawater. And so we used aquarium salt that sort of mimics seawater more than just putting sodium chloride like a table salt. So you can buy it in a nice, huge bucket that will be a lifetime supply of salt.
Perfect. Lee has asked,
and one more point to that. We did consider getting actual seawater from the ocean for this experiment, but the reason we ended up going with sort of the aquarium seawater was we just wanted to reduce the potential of bringing in, any additional potential pathogens or microbes that could influence it. But that's a great question. Thanks, Gavin.
Thank you. I've just switched question actually, because I've decided I'm going to ask. This is something you can do maybe during Michelle's talk. Jonathan has said, I live on an island. Jonathan lives on Malta, lovely Malta, which is generally saline. I'm going to say saline, and has alkaline pHs, and mycorrhiza were never studied. Could you please share with us the papers that you mentioned so that they can do similar studies? So perhaps while Michelle's chatting, obviously you'll be engrossed in what Michelle's saying, but if you could maybe put some links in the chat, that would be great.
Definitely.
Thank you so much. We'll give it a go with Michelle. I'm sat here like a Bond villain, just stroking the dog.
Thank you, everybody, for coming. Thank you for your patience. I am here to talk to you about some recent work that I have been doing on the golden oyster mushroom, which is this amazing golden oyster mushroom that has started popping up in a variety of places in the United States, North America, and also has started to show up a little bit in Europe. So I'm at the University of Florida in Gainesville, Florida, where I just started a lab, and I'll start my talk. So just to start with, I wanted to provide you all with a working definition of invasive species, because when we talk about invasive species, there are sometimes hot button words here, and so I just want to let you know where I'm coming from with when I say invasive species. So what I mean is something that's been introduced from an outside origin, something that is spreading, and something that is increasing in abundance. And I know Elena just talked to you about fungi. I wanted to step back a little bit and give us just a sort of broad overview of fungi very quickly. There are about 155,000 described species of fungi. That only represents about 5% of the fungi that are probably out there. So that means at least 95% are undescribed, and they occur in every ecosystem on Earth. The current estimates of the number of fungi range from 3 to 5 million species. And fungi are really cool and have fascinating lifestyles. And so you just heard about ectomycorrhizal fungi from Elena. Fungi can also be saprobes that decompose dead and organic material. They can be parasites that exploit living tissues, be they animal, plant, fungal, etc. They can also be symbionts that form really cool mutualistic relationships with mycorrhizae, like Elena just told you about, and also sometimes with other organisms like animals. And these aren't all of the possible modes. You might have, some fungi may have multiple lifestyles, and so they can really operate on a continuum from parasites to symbionts, which fascinates me immensely, but that's the topic of a different talk. So today, we're really focusing on decay fungi, and one decay fungus in particular, the golden oyster mushroom. But I want to first, before we dive in really to golden oyster, to also tell you about why forests need decay fungi. So there's a lot of carbon stored in wood. 70 petagrams is one estimate. That is the equivalent to the mass of over 700,000 aircraft carriers. Not a great vision right now, but that's equivalent to the mass of that. And the release of this carbon and other nutrients depends on decay fungi. And so these fungi are really, they're key decomposers in the carbon cycle, in the global carbon cycle. And I should say most decay fungi are in one fungal phylum, and that's Basidiomycota. There are many, many fungal phyla, as you might remember from that nice picture of the diversity of fungi, and most of these fungi are in one phylum. And so Decay fungi get divided up into two main groups. There's still a continuum. So, you know, if anyone's out there and like, oh, wait a minute, she didn't talk about gray rots. There's a continuum from white rots to brown rots. And brown rot fungi leave more lignin behind when they're degrading wood, whereas white rot fungi leave less lignin behind. And as they're degrading wood, brown rot fungi shuttle more carbon to the soil. And here we've got a nice picture of a brown rotter, Laetoporus. This is probably Laetoporus sulphureus, the chicken of the woods. And then white rot fungi return more carbon to the atmosphere when they're decomposing wood. And here we have a picture of golden oyster mushroom, which is a white rot fungus. And any mode that they have, be they white rotters, brown rotters, or gray rotters, decay fungi really function as critical members of diverse communities, right? And so understanding how decay fungi, and in particular how invasive decay fungi may alter wood decomposition is really critical. So there are a couple of examples of invasive wood decay fungi. So there's Serpula lacrymans. That's an invasive dry rot fungus. And its ecological impacts aren't widely known because it causes a lot of damage to wooden buildings across Europe, North America, and Australia. And that's really how people know it is this interior dry rot. And it's caused immense damage to wood-built structures, costing millions of dollars. And then there's, I'm going to butcher this name, Favolacea calocera, which was first documented as spreading in Australia and New Zealand in the 1990s. And this fungus there associates with about 50 species of plants, but as well it's associated with wooden shipping pallets. And it's since been introduced to Italy from the invasive New Zealand population. And then, I'm somewhat biased, but one of my favorite invasive wood decay fungi, which is a terrible thing, this weird thing to say, right, is Urpex subulatus, which is a beetle-cultivated decay fungus. So this decay fungus is also a white rot. It travels on these tiny ambrosia beetles, which are also invasive. And these beetles garden the fungus and feed it to their young. At any rate, this is another example of an invasive decay fungus, and in this case, a cultivated one. Interestingly, and maybe I called this Flavodon a moment before, it's Urpex subulatus. It's recently changed genera. Urpex subulatus in inoculated experiments has shown to be a strong competitor that changes fungal communities in wood. And so by doing this, right, a lot of definitions of invasion include harm. By doing this, this would cause an environmental harm. Okay, so moving from one invasive cultivated fungus to the next, we have the golden oyster mushroom, which I'll frequently be calling GOM. That's its new nickname. It's from Northeast Asia. It's edible. A lot of people really like to eat it. It's cultivated widely. It was introduced to the United States and to a lot of other places via commercial cultivation, likely through the widespread use of these home grow kits that are so widely available. And Andy Bruce at the University of Wisconsin-La Crosse did her master's thesis on this, and I think she finished in 2018. And she was the first person who really noted that these grow kits might be the reason that golden oyster mushroom escaped cultivation. And so when you see this mushroom, in the woods. It's really characterized by aggressive growth, a lot of large clusters of mushrooms, the rapid fruiting. And when we first started observing it in North America, it seemed to really like elm, at least where most of the people were observing it, which was largely in the Midwestern part of the United States, where there's still a good number of elms. But now we see it on a lot of other coasts. And so I was in Wisconsin at the time that this work was taking place, from 2022 to 2025. And our early observations in Wisconsin of golden oyster mushroom in the wild really included a lot of trees that had snapped and fallen, a lot of elm trees that had snapped pretty soon after death. And you can see on this picture on the left, this just clusters of golden oyster mushroom from the base. And then if you follow the trunk into the woods, the snap trunk, it goes all the way up to where the canopy was. And we were seeing it around and thinking, my gosh, these elm trees really normally would be staying on the landscape for 10 years after they die, but when they get golden oyster mushroom, they fall down pretty rapidly. And of course, right, with these large clusters of fruiting bodies, there were a lot of people harvesting them, walking out of the woods with garbage bags full of golden oyster mushroom to put on the dinner plate. And they are indeed like quite tasty. I used to enjoy them. I think I've had them in my lab too much. And after you smell a rotting mushroom enough times, you maybe don't want to eat it as much. But this is a batch of golden oyster mushrooms that I cooked up myself into a little, what I call gomlet. So given these observations and what was going on in Wisconsin and other places in North America, we wanted to know how golden oyster was spreading in North America and how quickly it was spreading, and whether or not it was associated with lower fungal diversity or different fungal communities in its invaded range. And so this work recently got published in Current Biology. You can scan that little QR code if you want to get a link. Unfortunately, it is not open access, so I can provide a PDF to John, and he can maybe send it out to people if people are interested. So I'm going to walk you through the spread of golden oyster mushroom in North America from 2016 to 2023. And these are data from citizen science, community science databases, iNaturalist, and Mushroom Observer. So this is a crop map of North America focused on the United States where GOM was first introduced. And the yellow states are states where GOM has been observed. And so once a state has an observation of GOM, it goes yellow. And then the little orange dots are the observations, which are non-cumulative. So in 2016, based on these community science databases, we had 5 observations of GOM in 5 states, right? And so we go to 2017, you can see we've still got the states colored yellow where it was observed. And these are new observations, new orange dots, 2018, 2019, 2020, 2021. You can see that one Canadian province has gone hot. 2022, we've gone down to the southeastern US. 2023. And this is where this map stops, but it is not where GOM has stopped. So I made a little map just a month ago of 2025 without the orange dots, but just with the states colored in yellow. But even since then, we have new observations in some of the southern states. You can see, and in more Canadian provinces, actually. And so you can see that the range is still increasing quite rapidly. When we go back to iNaturalist and ask how golden oyster mushrooms mushroom observations in the US compare to all other fungi observed in the US on iNaturalist, the rank has moved of golden oyster mushroom from non-ranked at all, right? Like 3,000th in the country all the way up to number 8 in the country. So the GOM observations per year have increased exponentially in the last 10 years. And that's, yeah, so that's what's happening. So hopefully I've shown you how quickly GOM spread in North America. And now to answer the second question, is GOM associated with lower fungal diversity in its invaded range? So for this, we used environmental DNA metabarcoding, which Elena has already explained to you a little bit. But if you're thinking about how many fungi are in wood or in water, as this picture depicts, or perhaps in sand, or if you take a swab of your belly button or whatever, right, you might do some traditional methods such as culturing, or even going out in the woods and looking at the number of fungi on a log, etc., or doing microscopy. Or you can do DNA-based techniques to get at all of the fungi in a sample, just using DNA and molecular-based techniques, PCR, and sequencing every single fungus in a sample. And so from this, we get it really depends on the sample type and the substrate, et cetera, but you might end up with sequences representing anywhere from 1 or 2 different species of fungi, all the way up to maybe 1,000 or more species of fungi in a particular sample. So for this survey that we did on trees with and without golden oyster mushrooms, right? With and without fruiting bodies, we took drill samples of the wood from these dead elms at 3 different heights. We collected the wood shavings and then did DNA extraction, PCR, and metabarcoding on those wood shavings. And we did excessive controls, including negative controls out in the woods, which if anyone out there does any kind of molecular work, you're like, wow, you are really into controls. You're not wrong. And so with these drilled wood shavings, right, we just took it through this molecular pipeline, did bioinformatics. We did Illumina sequencing, just like Elena did. And here's Aishwarya, the first author of this paper, doing her lab work for this. So what did we find? First, we found that trees that had golden oyster mushroom in them had many fewer other species of fungi in them, lower fungal richness. And so on this plot, you have operational taxonomic unit richness. That's roughly correlated to species, as Elena said. And in this golden bar on the left, GOM detected, you can see that is lower than where we did not detect GOM. And this is a different kind of way to visualize fungal communities than the canonical correspondence analyses Elena was walking you through. This is a non-metric multidimensional scaling plot, but think about it the same way. Dots that are close together have more similar fungal communities than dots that are further apart. Each dot here represents a wood sample. But you can see where we had golden oyster mushroom detected, you have a very different fungal community than when you do not have golden oyster mushroom. But what this means is that GOM is correlated with different fungal communities, with different species richness, and it seems to be competitively dominant given these correlations, and could perhaps be displacing existing fungal communities in wood. What is also interesting here is that this these results with non-inoculated trees of an invasive cultivated mushroom, human cultivated mushroom, bear a striking resemblance to results that we saw previously with this beetle cultivated invasive fungus, Herpex subulatus. Something that I would like to pick up somewhere down the line, but am not going to discuss much more now, except to say, my gosh, look at that. We have really similar things with these cultivated invasive fungi. And I should note too, with this Herpex study that is on the left here, the beetle one, these were inoculated trees. To see such a striking resemblance in non-inoculated trees with golden oyster mushroom means this thing might really be quite competitively dominant. So the study that we did shows you that we have an expanding range, growing populations, reducing fungal diversity. This is what we know about golden oyster mushroom. There's still a lot we don't know. So first, we don't know if golden oyster mushroom is a pathogen. We don't know when it gets into a tree, when the tree is living or when the tree is dead. We don't know anything about its life cycle. So some wood decay fungi are present in a tree prior to tree death, and these are called endophytes. And you've probably heard about some of those from Emma Gill-Martin or Lynn Boddy. And other wood decay fungi enter a tree once it's dead, perhaps, or start fruiting once it's dead. We don't know how long until golden oyster mushroom fruits or becomes reproductively active. So to answer some of these questions about when fungi get into trees and how we detect this, Megan Dudenhafer at the U.S. Forest Service Center for Forest Mycology Research has been working on some new molecular techniques for detection of golden oyster mushroom in trees that don't have fruiting bodies in them yet, because currently our detection relies on seeing these mushrooms or getting wood from trees and sequencing it and culturing it without even knowing if golden oyster mushroom is there, which takes a lot of time and resources. None of these techniques are rapid, and so we really need to have new techniques for detecting emergent invasive species. So we have this new quantitative molecular assay that Megan is in the process of writing up. And we also don't know how golden oyster mushroom is spreading. Like, well, it's got these giant fruiting bodies, these giant mushrooms. And if you look at them, they're spreading spores, like they're dropping spores readily. But you also look at it, you see these mushrooms are going from the base of the tree all the way up to the canopy of the tree, suggesting it may have some help in multiple entry points in a tree. And so to look at this, we have I'm going to say little study, but you'll laugh when you see these pictures, but we're looking at whether or not there may be some potential other vectors of GOM. So we cut down living elms, and this is us working in the woods to get the elm trees down, have set up a field experiment in my collaborator Mark Bannock's tree farm, where we have different treatments of these elm logs to see if they get golden oyster mushroom, and none of them are inoculated, right? This is a 2-year study to see what happens. And you can see, we're starting to see some things fruit on this, and we are doing DNA-based techniques on these logs periodically to check. We'll be taking down this experiment, I think actually later this year. And so, stay tuned. You can see some of these logs have little beetle holes on them. Some of them are generating all kinds of other fungi. And we know that they're in a place where golden oyster mushroom is, so they may indeed become inoculated with it. So, right, I'm not gonna take you through little bits and pieces of all the rest of these, but I'll just say, we're looking at preferred hosts. We have preliminary tests underway. Whether or not GOM is actually out-competing native fungal species is something we really hope to start on very soon, to actually do that test, because right now what we have is correlative evidence and so many other questions about this fungus. The other thing is, this fungus is still widely cultivated. You can still buy these grow kits. You can have them shipped to you online. As I was getting ready for this talk, I looked to see how many different places were selling golden oyster mushroom in the UK, and it turns out there's a lot. You can still buy it here in the farmers markets and the grocery store, and you can buy grow kits, but some companies have recently discontinued these grow kits, those that are intended for outdoor use, and that is that's lovely until we can figure out a way to stop the fungus from escaping the grow kits. I am appreciative that some companies have decided to discontinue some of the kits that are intended specifically for outside use. So aside from discontinuing selling some grow kits, which is not going to do a whole lot right now, admittedly, what can we do? We can work with mushroom farmers. For golden oyster mushroom, the genie is sort of out of the bottle, right? But at least it is in North America. Maybe it's not in Europe. And there's growing interest in mushroom cultivation. And so maybe we can start to think about how many cultivated mushrooms are native and how to perhaps grow more native mushrooms to stop this from happening again. And think critically about what we can learn from golden oyster mushroom. You know, maybe it's as simple as how growers compost spawn, right? And that's not saying, you know, don't grow anything. It's just saying, oh, perhaps after you're done with your spent spawn, we can work with you to develop ways to dispose of your compost so it doesn't keep, so the mushrooms don't keep re-emerging from the spent compost, which they do sometimes. What can you do though? You could help document golden oyster mushroom in non-cultivated settings. Community science platforms are wonderful. We used iNaturalist a lot for this work. I know there are others, and I'm very keen to know which other community science platforms folks are using. We also use Mushroom Observer, but there aren't as many observations on that. And even if you cannot provide a good ID of the fungus, you can take a picture, do the recording. You know, other folks will provide IDs and weigh in on that and make it right, research grade, etc. The data are an excellent record. And if you can, even if you cannot provide an ID of the fungus, if you could note the type of tree, I know that is very hard, but if you can, that is wonderful. And if it is alive or dead, is really really critical information for us right now. We're seeing golden oyster mushroom pop up occasionally on living trees, but most of them appear to have dead parts. And so as we're trying to piece together when it's coming into a tree, that helps a lot. And so this is a screen capture from iNaturalist from this morning. You can see that the fungus has spread quite a bit. These are, and I should say this is using all observations, not just those that are verified. And it's starting to pop up a lot more in Europe than it was even just 6 months ago. And one observation now in Australia too. But I think the observation in Australia is from a cultivated grow kit. So we'll keep the hope alive on that one. Okay, I have a lot of people to thank, and thank you all for listening and bearing with me through some technical difficulties. Yes, and especially my co-authors and collaborators, all listed here. Again, the QR code for the paper, and yep, thank you.
Thank you very much, Michelle. That was wonderfully terrifying. Thanks. Have we got Elena? Do you want to come back as well? He says into the void. Yeah, there you are. Fantastic. Thank you both. Two brilliant presentations. Really great. Lots of questions, lots of appreciation in the chat. As always, we will send the chat and the questions to our presenters so you can have a look at it. And then if there's any questions that particularly dazzle you, you are free to respond to them. Where are we? Quarter past. So we've got about 15 minutes for a few questions. So which ones did I prioritize? So this is going to be a depressing one, but I'm going to go for it. It's one for Elena from Jocelyn. Jocelyn says, like so many ecosystems, how far are we from being too far on the tipping system to mitigate the loss of this one? Are we doomed because of salinity?
Yeah, that's a great question. So the question, just to reiterate, is, you know, how do we know when all hope is lost? You know, it's a great question, and I think it's sort of ecosystem dependent, but I think sort of what the results of my work sort of suggest is that, you know, certainly in those higher elevation areas, you know, where the soil salinity is much less high, you know, those could be sort of areas to focus more on for restoration. So in some of the lower lying ecosystems that we know will over time be more and more impacted, maybe we just focus on those higher elevation sites. So I like to think that not all hope is lost. There's really importance in trying to preserve areas. I mean, even just creating stepping stones, right? Like creating smaller fragments of forest can really help. But yeah, in terms of all ecosystems, it really is dependent on the area.
Fantastic, thank you. That seems vaguely positive. We'll take that. We'll take that as a little win. Michelle, this might be a chance for another positive answer. I don't know. Laura has asked, in the hypothetical situation where a fungus like golden oyster was noticed in a new country at an early stage of invasion, is there anything that can be done to stop the spread? Laura says, I imagine it's more difficult to control than other invasives.
Yeah, that's a great question. It is possible that stopping the spread early on could simply be that you could stop the spread early on simply by ensuring that the mushroom was not released into the environment anymore. Unfortunately, though, like as Laura suggests, it is more difficult than that, because by the time we notice the fungus fruiting, it's perhaps already been in the wood for maybe even years. So it's a little bit of a conundrum with a decay fungus that is perhaps persisting in these trees before it emerges. But that doesn't mean that we shouldn't try.
Absolutely, thank you. And I'll come straight back to you, Michelle, because there's another question, sort of similar, I guess. But Anthony has asked, what is golden oyster mushroom's fungal competitors in its native range? And Anthony appreciates that it's heretical to suggest introducing them as well, but are there similar native fungi that may be expected to compete eventually?
That's a great question. I wish that I knew the answer about what the competitors are in the native range. I really would like to go to the native range actually to sample and to see this thing growing. Anecdotally, it seems like in the native range, like so many other invasive fungi, I'm thinking in particular of like Amanita phalloides, for instance, which is invasive in certain parts of North America, but not in its native range. The appearance of the mushroom seems to be quite a lot more ephemeral in the native range. And so, right, like this natural enemies hypothesis and why, right, like, or enemy release, like why is it invasive in one place and not the other? What is keeping it in check? And it may be more than just, right, the competitors. But I would say insufficient data to answer the question about the native range at the moment, but it's a really great question. And insofar as what might be able to keep it in check here, that is something we're actively trying to look at. And based on what the DNA data that we have right now, there are a couple of different fungi that really seem to coexist with it well, including what was Polyporus squamosus. Druid saddle is now I can't remember the genus, but yeah, so we're starting to pit these things against each other on plates and in wood blocks to start to try to get at some of those questions.
Thank you. Elena, Paul's asked, or Paul said, first of all, that fungi that help with salt tolerance could be a useful tool, especially on roadside areas in the UK, because we do a lot of gritting and salting over here. The only issue is the impact that could have on the current microbiome with potentially invasive species. And Paul's asking if you've got any suggestions as to how we could overcome that. You know, can we use that as a way of helping trees for salt damage? And how will we overcome those problems of impacting the current microbiome?
Certainly, yes, that's a great question. And depending on sort of where I give this talk, because I'm located in the northern part of the United States, de-icing salt is a real concern up here. It's certainly not a concern in Florida, but you know, that is a concern up here. And many highways have, you know, pine trees and oak trees and all sorts of trees that are right alongside the highway that exhibit salt stress. So you'll get trees that have really browned out after winter desalting. And so some of the work that I didn't discuss that was early on in my PhD was sort of thinking about restoration efforts and outplanting trees or seedlings that have mycorrhizal fungi associated with it. And that whole chapter and paper was, you know, looking at the difference between getting commercial inoculum versus using inoculum in the environment. And so that's always my suggestion, is to try to keep your microbiome communities, you know, close to home. And so for a lot of these fungi, there might be fungi that are more salt tolerant that are naturally existing, but perhaps they're being outcompeted by other fungi in the system. And so I would say if you could somehow, you know, do some studies to look at the types of fungi you have in your area and screen them for salt tolerance, and then perhaps use those to inoculate and outplant, or just select higher salt tolerant plants that can exist closer to roadsides. That's another way to kind of mitigate this this salt stress. So that would be my suggestion.
Thank you. There's a couple more questions I'm going to ask, but I'm going to ask a question from myself first, actually, because you both described some pretty serious issues, and I'm just kind of curious, like, are people taking this seriously enough? Like, are you finding you're just talking to groups like we've got here, which is people who are already interested in this theme, and then they're sort of learning and taking it on board? Are the politicians listening? Are the policymakers listening? Is this, you both described things that really should be higher up the list of priorities than I suspect they are. And I was just wondering what luck you'd had in actually talking to people about this. Elena, if you want to go first.
Yeah, definitely. So it's sort of along the lines of the de-icing salt. I mean, I think, you know, the Florida pine rocklands is a really unique ecosystem, although, as I mentioned, there's a lot of ecosystems in North Carolina and Georgia, Louisiana, you know, all sort of along the Atlantic coast and the Gulf Coast that are suffering from things like ghost forests and salt intrusion. But where I'm currently located, the de-icing salt is a real concern. And I think certainly the scientific community is taking it very seriously, but I think for a lot of folks, once they have the information, they are really interested in how they can help. And so things like, how can I reduce how much salt I'm putting on my sidewalks in the winter, and how does that influence things? Because as we know, once salt gets into the soil, it's really, really hard to remediate out. And so I think just being more conscious about the things that we can control, right? Things that are larger, sea level rise and stuff is sort of harder for one person to combat. But I think folks are interested in what are the things that I can do that help reduce salt input. And that can be things like reducing how much salt you put on the sidewalk and stuff. So I think people are taking it seriously. I think, like many folks, they want tangible things that they can do to feel like they're helping with the problem.
Thank you. And Michelle, same question to you. Are people alert to this issue? I mean, in the States, is this something people are aware of? Are behaviors changing because of the work you've been doing?
Well, so since the current biology paper came out, it was released online in July of 2025. We've had a massive response from the media, like absolutely massive. A lot of people found out I was moving from Wisconsin to Florida via the New York Times. So that has been good in putting the issue out into the limelight, but I've not seen anything move much past the flash pan event that happens when you publish a flashy paper on a very, shiny yellow mushroom. And except, right, seeing North Spore discontinue some of its grow kits, those meant for outside, you can still buy the inoculum in syringes and things like this, right, for lab-based operations. I have not seen a lot more happen. I've had a few composting companies and places like that reach out to me to say, what can we do? Can we start to think about how to mitigate this? But policy has not changed, and we are trying really hard to get more research off the ground to help sort of work with growers and composters and people to sort out what can we do to stop this spread but the short answer is there's not anything we can pinpoint right now that has exactly happened. I mean, I can still go out in the grocery store and the farmers market, et cetera, and buy golden oyster mushroom. There's been a lot of pushback of, well, I really like to eat this thing. Why are you telling me it's invasive? So we're trying to work through that and keep working with the folks who have reached out to try to find some creative solutions.
Mm-hmm. And Christine said in the chat, homegrown kits should have instructions on how to dispose of them once they're exhausted. I don't know if that happens or not.
They absolutely 100% should. Absolutely. That would be such an easy thing to include. And I don't know if that's included or not either. I've not had a homegrown kit we used to buy a couple in the lab to generate spores for experiments, but we've not had one for a little while. And so perhaps that is something that's being done, but also would be a great, easy thing to reach out to them and do quickly.
You haven't bought one since you got those ones you chucked out in the woods when you were done with them. Elena, did you have your hand up there? Sorry.
I just wanted to say one more thing to answer this question, which is, I think the central theme between Michelle's work and mine, even though the topics are vastly different and scary in their own way, is for both of us, there's just so much that's unknown about these systems. And so I think, that's just my plea for always the need for more basic research and support for basic research, because it really is important in sort of having actionable steps. We really need to understand these systems before we can make management recommendations. So that's all I'll say.
No, absolutely. Thank you. We've got loads more questions. We haven't got time to work through them all. So as I say to everyone out there and to our presenters, we'll send you all the questions afterwards. And then if you see anything that particularly dazzles you, you can reply to them somehow. We can figure that out. So you'll have the opportunity to do that. So sorry if I didn't ask your question, folks, but I do, as always, have time to ask my final question, which is, what is your favorite tree-related book? And I'm going to go to Elena first.
Yeah, this is a great question. And I feel like I'll only answer this question if I'm promised that I can get the book list that you've been generating, because I'm an avid reader. I love to read. And so I'll only answer if I have a pinky promise that you'll email me that list.
Pinky promise. It's available in our ad magazine and it's probably online somewhere. So I will get it over to you. I promise. You'll be added to that list.
Okay. Excellent. Well, mine is, when I was thinking about this, I thought, well, there's a sort of nonfiction related book that I could suggest, but I'm going to go a little bit unique and say that there's a fantasy book that I really liked called Uprooted by Naomi Novik. And it's basically about this, it's about a forest, which they call the wood, which is, sort of enchanted and a little bit horrifying. But there's sort of I won't spoil it, but there's a larger history to that wood. And so I kind of like that it's forestry meets fantasy. And so that's my recommendation.
I like it. And that is unique. Nobody said that before. Oh, everyone else has disappeared and it's just me.
No,
can anyone hear me? You can hear me. It's all changed and it's just a big screen and I can't see anyone. But as long as you can hear me, that's fine. Right. Okay, fine. Anyway, I'll put that in the chat. Michelle, what's your answer?
So mine is less interesting than Elena's probably. The Fungal Decomposition of Wood by Rayner and Body. And the reason is that I came to fungi and specifically to decay fungi by way of birds. And this was a critical book for me as I was just learning about wood decay when I was a PhD student. And yeah, that's what I'll say.
I like it. That's another new one. We've not had that one either. So you've added new books to our list, which is always really appreciated. I chucked a link in for Elena's. I haven't yet chucked a link in. I can do that. I just go for random links. Other bookshops are available and I'll put it in there. There we go. Boom. Right. Thank you both so much, not just for your book selections, but for two wonderful, thought-provoking presentations. It was a pleasure to have you both on. Thank you. And thank you to all of our audience, of course. You're all wonderful, most of you. And thank you for coming. And hopefully we will see you next week for Jim Chambers talking about ancient trees and definitions and things of that ilk. So please do sign up for the webinar next week and all the webinars in our series. But once again, I would just like to say thank you to our two speakers tonight, Michelle and Elena. Absolutely brilliant. Thank you for joining us and hopefully see you again soon. So take care, everyone. Stay safe. Bye-bye.